So Maya and I will be presenting the International Brain Lab's brainwide map dataset, how to access the data, and how to use our Python API that's called ONE. So first, I'll give you an overview of the brainwide map dataset itself, what data were acquired, how the data are organized, and then how to access the data using our API. And then Maya will go over one of the analyses used in the brainwide map paper, and how to replicate that with the dataset and API. So why collect a brainwide map dataset? As I guess all of you know, there's neural patterns generated across many different brain regions during behavior. And so stands to reason that to properly understand how those groups of neurons produce behaviors, makes sense to record from the entire brain during a given behavior. So the behavior we chose was two alternative forced choice decision-making task that required the integration of sensory information with prior contextual knowledge in a visual task. So a few things that can be potentially researched with this dataset include how sensory information flows across brain regions, where prior information is stored, and how past actions influence behaviors, as well as how neurons encode task-relevant information and produce movements. So let's give you an overview of the dataset itself. This is an image of a mouse brain depicting all of the insertions made over 12 labs in nearly 140 mice. So this comprises many sessions and almost 700 probe insertions. So the task itself, it's a head fixed task. The mouse sits in front of this screen, in front of a couple of cameras, and under its paws, it has a little Lego wheel. And so after a period of enforced quiescence, where the mouse has to keep that wheel still, a stimulus appears on the screen. It appears on either the left or the right-hand side of the screen, accompanied with a little beep. And the mouse has to move the stimulus via the wheel to the center of the screen. So if it manages to move the stimulus to the center of the screen, it receives a drop of sugar water. If it moves it too far in the wrong direction, there's a timeout period and a white noise burst. So there are two main task parameters that vary across trials. First is the contrast of the stimulus, visual stimulus. And this includes zero contrast trials where the mouse has no sensory information about which direction to turn the wheel. And the second parameter that varies is the probability that the stimulus will appear on the left-hand side. So this varies in a blockwise pattern, alternating between 80% likelihood on the left to 20% likelihood. So here I'll go through some of the hardware devices used to make up this data set. First one being the behavior. So we acquired the behavior with Bonsai and some hardware called BPOD. The raw behavior data typically stored in a folder called raw behavior data. It undergoes some pre-processing, which involves extracting some interesting trial event times, task parameters, and then also the wheel position and timestamps. And these are synchronized to a common clock. And I should say the pre-processed data is stored in a folder called ALF. I'll go into more detail with that in a moment. Second set of devices are the cameras. The raw data are stored in a raw video data folder. We have typically 3 cameras. And then the pre-processing for this involves feature extraction and tracking with Deep Lab Cut, as well as some motion energy extracted from some regions of interest, such as the whisker pad there. So again, these extracted features are stored in an ALF folder. And then the final piece to this is the NeuroPixels data. So this is stored in the raw EFIS data folder, and the pre-processing includes feature extraction and also some spike sorting with pi-killer sort, and also later on we used another sorter called IBL sort. So this produces data sets including spike times and cluster IDs and things like that. So once this was done for each experiment, all of the data were then copied to a remote server, and information about the data sets were uploaded to a metadatabase that we call OpenAlex. So that dataset information includes which files were associated with which sessions, file hashes and file names and such like that. So now I'll go into a bit more detail about how these data are structured on disk and how that relates to the API we use. So a session sort of looks like this on disk. Each session is uniquely identified by a specific session path, which comprises the subject name, which in this case is DY14, as well as the date and the session number on that date, starting from 1. So this path uniquely identifies an experiment. But on the database, we also have UUIDs to identify the experiments, which we call EIDs. Second, we can have 1 or 2 probe insertions for each EFIS experiment, depending on whether it was a unilateral or bilateral recording. And the data for these, both raw and pre-processed, are stored in the probe 00 and probe 01 subfolders. By the way, can people see my mouse?
Yes, we can see.
Okay, good.
If you have a laser pointer, it might be even better.
Well, I don't know how to do that, so.
No worries.
Yeah, so as I mentioned, multiple probe insertions can be recorded per session. And likewise on the database, each insertion is associated with this long UUID, but can also be identified uniquely with a combination of the probe name and session ID. So I mentioned these subfolders. These subfolders are referred to as collections in ONE. And the main collection of interest is this ALF collection, which stands for Alex file, because this contains all information that's been synchronized and pre-processed. So all data within the ALF collection, all the timestamps and event times are in seconds relative to the session start. And then also it's just worth mentioning that not every session contains the same data. So training sessions won't have any EFIS data, for instance. The number of probes varies, and for various other reasons, there may be missing camera data or other such differences. And so the final part of the sort of data structure on disk is the file naming itself. So data sets all comprise 3 parts: an object, an attribute, and an extension. Objects are basically the first part of each file name, and files within a session and a specific collection will always have the same leading dimension. So in this case, the object for the body camera is called body camera. And if you were to load bodycamera.times or bodycamera.dlc, you can be confident that the leading dimension is frames, and they'll both have the same number of frames in the file. So next I'll just go through how we can access these data using the Python API, which we call ONE. So ONE has 2 principal modes, so to speak. The first is a local mode, whereby you can simply list, search, and load data from disk. So I've just shown you how the data are organized on disk. And this allows you to both share data and also load data using various filters such as, object name or subject or date, etc. But if you provide ONE with a URL to our OpenAlex database, you're also capable of searching the database itself for all of the brainwide map data, as well as other IBL data, in fact. And so with this, you can load the data and ONE will download it for you if it's not already on disk. So let's go through some of these methods. The first method is the search method, which allows one to search over sessions given any number of filters. So, for example, searching for sessions for a given subject. Another example would be brain region or sessions containing particular data sets. However, as I've shown you, we do have bilateral recordings. And so if you want to search for data in a given brain region, it makes sense to search over insertions instead of sessions. So for that, we have this search insertions method. And this returns you a list of probe IDs instead of session IDs. So once you have an experiment of interest, you can then explore the data using this list datasets method. What this does is you provide it an experiment ID or a session path, and it just returns you a list of the data available for that session as paths relative to the session. And then finally, the loading of the data. There's several methods for loading the data. First one being load data set. So for this one, you provide it a dataset name and an experiment ID. And if there's multiple datasets of that name, you also have to provide collection, i.e. the subfolder containing that dataset. And it will download the data for you if it's not present on disk, and then load it into a NumPy array. There's also a list data sets method, which does the same thing, but you provide it a list of data sets. And you can also have it just return none if that specific data set doesn't exist for that session. And then finally, we have load object. So as I mentioned before, the object is the first part of a file name, dataset file name. And given that they're guaranteed to have the same leading dimension, it can be loaded into tables in Python. So in this example, we have a clusters object with multiple different data sets representing that object, such as clusters.amps containing amplitude, clusters.depths, UUIDs, etc. So to load a clusters object, you pass in the experiment identifier, the object name, optionally the collection, and it returns you basically a dictionary of attribute keys to the corresponding NumPy arrays. And these dictionaries can then be easily converted to a pandas data frame. So I mentioned experiment IDs potentially being a session path or a UUID. Every list and load function can take either as an input, but they can also be interconverted with these two methods here, the EID to path and path to EID methods. And likewise, probe insertions can be converted between experiment ID and probe ID using either EID to PID, where you pass in an experiment identifier and it will return you a list of the probe IDs and their names, or vice versa, basically. Okay, so now I'll pass you to Maya, and she'll go through one of the brainwide map analyses.
Yeah, thanks, Miles. So yeah, in the second half of this talk, I'm going to introduce you to some of the analysis that we've done on this brainwide map dataset as part of the IBL. And we're also going to go into detail into one of these analyses and show how you can use the tools and also the data available to be able to replicate this. So if you, so after we collected all of this data within the IBL, there's a task force that was designated to try and make sense of all this data. And the idea is that we wanted to understand which areas in the brain were processing different aspects of the task. So the way that we approached it was that we decided to, sorry, can you go back? The way that we approached it was that we decided to split the task into different variables. So for example, visual stimulus, feedback, choice, and then apply a number of different analyses on time windows of interest that correspond to each of the different task variables. So the analyses that we applied included encoding, decoding models, single cell statistics, and also trajectory or manifold analysis. So this figure here shows one of the examples of the mouse, sorry, one back, shows an example of the results for choice, the choice variable. So this is when the mouse decides, the direction that the mouse decides to turn the wheel, so the visual stimulus, so either to the left or to the right. And in this case, the time variable or time window that we were interested in was the time prior to the first movement onset. So this is the time window prior to the point at which the mouse decides to make a choice and move the wheel in one direction or the other. So in these 4 or 5 plots at the top, we show the results from 4 different analyses. And these basically show the results on a flattened representation of the brain. So we've taken these from the Swanson flat map. And these are quite nice displays in that they allow us to view all of the data in different brain areas on one single plot and allows us to interpret data across different analyses in a quite concise way. So in these results, we can see that darker areas correspond to more significant activity. And for the variable choice, what we found was that in general, it was the areas such as the hindbrain, midbrain, and cerebellum that actually encoded choice most strongly. So I'm not going to go into any more details of the results per se. So I recommend you to have a look at the preprint, which we've linked at the bottom of this slide. And this will have all of the results and also information about the different task variables as well, such as the visual stimulus, the block, and which areas in the brain were involved in these aspects. So if you can go to the next slide. So I want to focus in on one analysis in particular. So this is a trajectory analysis, and the results highlighted here show the results for this analysis. So this is a dimensionality reduction technique where basically we use PCA, or principal component analysis, to take our high dimensional data and reduce it into a lower dimensional space. And in doing this, we could basically choose the top, for example, 3 principal components that described our data, and view how the trajectories of these principal components varied between when a choice was made on the left, so the data for when a choice was made on the left versus when a choice was made on the right. So that's what you can see in the top right figure here. So we have 2 trajectories in 3D, in kind of 3D coordinates, the right choice, which is shown in red, and then the left choice shown in blue. And this basically shows how the activity diverges for these principal components up to our time point of interest. And in this case, t = 0 indicates the first movement time. So what we can do is actually compute the distance between these 2 trajectories in this kind of multi-dimensional space. And we can summarize that as just like a graph over time of how much these 2 trajectories diverge. So in some instances, you can see, so this is shown in the bottom right figure. So in some instances, you can see that the line's pretty flat. So in these cases, there's not much difference between the 2 trajectories between the left and right choice. But in other cases, there's a very marked difference. So particularly you can see for this region GRN, so the gigantocellular reticular nucleus, we see a very distinct difference between the 2 trajectories. So on the next slide, we show basically the full workflow, or full kind of analysis workflow that you can do to replicate this analysis. So the way in which we can do this trajectory analysis is the first thing that we need to do is get the spike sorting data. We need to find the regions, the neurons that lie in our region of interest. And so for the examples that we're going to go forward, I'm going to always refer to this region of interest as the GRN, because that was the area where we found the significant, very significant, like marked difference between the choice variable. So what we need to do is take the activity for each neuron and we compute the PSTH, or the peristimulus time histogram, around our event of interest, which in this case is the first movement time. So for each unit, what we do is we get like an averaged activity of how this neuron responds around this fixed time interval. So we get one for the left choice and one for the right choice, and we then repeat this for all of the neurons that we have in our region of interest. And in B, what we do is we basically stack these individual traces into a large high-dimensional array where we can have, where we represent all of this, the PSTHs for all of these neurons that we have in our region. So in C, we then compute a PCA on this. So we basically take this very high-dimensional array and take it to a lower dimension and just restrict the analysis, for example, to the first 3 components. Which describe the most variance in the data that we see. And we can plot these and we get these 2 trajectories. And as I was describing earlier, we can, in D, we basically compute the distance between these trajectories. And we can define a certain significance level at which we decide that the kind of displacement between the 2 trajectories reaches a significance level. And we can find the latency at which that point happens. So in E, we're basically showing the distance between the 2 trajectories, and the point marked as a diamond is when this difference reaches our defined significance level. And then this region GRN, around, I'd say 30 milliseconds prior to the mouse actually moving the wheel, we see that the decision has already been made. So the choice to move the wheel left or right has already been made. Okay. So in this next section, what we're basically going to do is use this analysis that I just introduced and show how we can use the data sets that we have, the API that Miles introduced, so the ONE API, in order to replicate this analysis. So I want to just emphasize that the aim is not to show you how to do a PSTH or a PCA, but more to show you how you can frame your question and leverage the resources that we've made available to get the data that you need, and then be able to do either this or another analysis that you can do with our data. So this will be split into, so we have 6 steps basically. So first I'll show you how you can install all of the necessary packages that you need. So hopefully if you've been following along with the assignments, you should already have all of these packages installed. Then we're going to use the search methods that we introduced earlier and find sessions of interest for our analysis. We'll then explore which data sets we want to analyze. So given the question that we're posing, which data sets are most relevant for our analysis. We'll show how you can load in those individual data sets using the documentation that we have. We'll then focus on just a single session and show how you can basically frame your analysis and extract the data that you need, and then finally extend this to all of the insertions that match our search criteria. Okay, so first, installing. So the thing that you'll need to do to start using our data is basically install this API, this ONE API that we were referring to earlier. So this can be installed just as a Python package. So you can just do pip install ONE API. And we also recommend installing another package called IBL lib. And this has a number of useful kind of utility functions that we've developed that just aid in some of the processing and also some of the loading. So the very first time you use ONE on a computer, you'll need to configure it. So this basically sets up a connection to that metadata database that we were referring to earlier that has all of this meta information about the sessions. And for the very first time, you need to just send in like a username and password. Once you've done that once, you should be good to go. And then every time you then want to do your analysis with IBL data, you just need to import the ONE instance and then instantiate an ONE instance. So next, we want to find sessions or insertions of interest that we want to use in our analysis. So this is where that searching kind of methods come in. So I want to emphasize 2 useful search methods. So they are one.search terms and also help one.search. So these you can just type into your Python terminal and they will output these kind of lists. And these basically show all of the keyword arguments that you can pass into your search methods. So here, for example, we have the session, the subject, the dataset type, and help one.search basically gives you more information about each of these individual keyword arguments. So it will describe to you what a subject is or what a data set type is. So for our analysis, the 2 keyword arguments that we're interested are the tag. So this basically corresponds to a group of data sets that we put into a data release. And in this case, it's this brainwide map data set. And the second keyword argument that we're interested in is this ATLAS acronym, because in this analysis, we're interested in just sessions or insertions that target the specific region, the GRN. So we want to basically pass that in as a keyword. So once we know the search, kind of the keyword arguments that we're interested in, we can then formulate our search. So in this case, because we're looking at electrophysiology data and we want those that pass through a specific brain region, we want to do our search on the insertions. And this is because, for example, as Miles was saying, for a single session, there can be multiple probes. So if you search on the session itself, it means that you'll be returned both probe 00 and probe 01 in your search. But perhaps it could be that probe 01 is targeting somewhere else and it doesn't actually have any neurons in GRN. So in the case of this kind of when you're targeting a specific brain region, this is probably the recommended way to actually start your searching. So we can pass in these keyword arguments into the one.search insertions method. And what we're returned is basically a list of probe insertion IDs. And in this case, we have 17 probe insertions that match this criteria that we're interested in. So at the bottom, we just basically take the first one in this list, which we're now going to use as our example session to work through before we then extend it, our analysis to loop over all of these insertions. Okay, so next we need to find what data sets we're interested in. So what data we need in order to start our analysis. So if we just recap what our question was, basically we want to compute the PSTHs for a bunch of different neurons in our GRN. And we're also, and then to then perform the PCA. So for this, we're going to need the spike sorting data, but we also need the trials data. So we need the data that has information about the each trial event, the choice, and also the first movement times. So the best thing, or what I would recommend doing first for your chosen session is basically just to list all of the data sets that are available. So one thing that you need to remember is that the list and load methods only work with the session EID, so not the probe ID. And since we started our search on the insertions, we need to convert them first using these converters. So from the PID, we can get out the EID, the session ID, and also the probe name. And we can then pass that into our one.list datasets method. So what you'll be returned is this long list of all of the datasets associated with the session, and you can look through them and see which ones correspond to what you think you'll be interested in. So in this case, we have some that have trials in their name and also spikes. So we have already an indication of what datasets we might be interested in. So then I would then refer you to look at this documentation, this Google documentation that we have called IBL dataset types. And this is basically a big reference of all the datasets that we've made available. So there's an index at the front that lists all of the individual datasets. And for each one, you can click on it and it will take you to the actual description of that dataset. So for example, on the right-hand side, we're describing some of the spikes dataset. So there's a spikes.amps, and it shows the units of that dataset, the expected dimension of that dataset, and also a short description of what that dataset contains. So you could also start by scrolling through this document, seeing what's available, and then you can then already get a handle of the kind of datasets you probably need for your analysis. One other useful method is this one.describe dataset. So this can be just launched from a command line or from a Python terminal, and once you have your ONE instance instantiated. And this gives you out information about a particular data set. So if you know the dataset name that you're interested in and you just want to find out a little bit more information, you can just pass it in and then it will give you a full description of what that dataset contains. Okay, so now, yeah, we have our insertions and we figured out which datasets we're interested in. So now we need to think about how we're going to load them in. So this will be using those one.load methods. But the first thing that I recommend is to visit this IBL loading data documentation page that we have as part of our IBL documentation. And there's this loading data page, and it basically has a list of these different notebooks that show you how to load in all sorts of different types of IBL data. So for example, the first one is loading trials data. We have one for loading spike sorting data. And I should also say we have not only EFIS data available, we've also got imaging data. So there's wide field data, fiber photometry data. So if you're interested in those kind of data sets, you can also see how to load them in using this documentation. So if you click, for example, on the first loading trials data, it will launch a page as you see on the right. And it tells you all of the relevant data sets that you'll load in. And the first kind of code snippet is the suggested way for how you can access that data. So in the case of trials, we suggest using this load object method, and you can just basically then load in the trials dictionary that you need that contains all the information about the task. One back, sorry. Yeah. Yeah, so okay, this is, I guess, the same. So using the documentation, it shows that for trials, we just use the one.load object. So we can then, in our Python console, just load in the trials for this session. And then on the next slide, if we also refer to the spike sorting documentation, you'll find that for that, the recommended way is not to use just an ONE method, but actually it's to use this spike sorting loader class. So this is a class that we've written in IBL lib, so that additional package I was mentioning to you. This basically uses ONE in the background. So it abstracts out some of the complications of joining together files, getting the correct files that you need, and it returns you kind of a nice set of spike sorting data that you can then go forward and use. So it returns you the spikes and also the clusters and channel information about where those clusters lie on your probe. Okay, so now we've loaded in our data, we can then go about exploring a little bit and setting it up for analysis. So in the first instance, we're going to explore the trials data. So as Mal said earlier, we can just simply, because this is a dictionary that's returned, we can just simply print out the keys and we can see what different objects we have in our dictionary. So in the question or the analysis that we're doing, we know that we're interested in two trials variables. So the choice, so whether the animal moved the wheel left or right. And the timings that we're interested in are these first movement times. So the first time at which the mouse moved the wheel. So we can extract those two variables, and what we're basically doing in the bottom of this, the bottom code snippet here, is splitting our trials according to these two criteria. So the ones where the choice was equal to 1, so where a left choice was made, and then when the trials is equal to -1, so when a right choice was made. And what we're also doing is extracting the timing of these events by indexing with the trials first movement times. So for the spike sorting data, we also need to do a bit of preparation. So one thing to note is that when you download the spike sorting data, it gives you the spikes and clusters from all of the neurons that have been detected by the spike sorting algorithm. So this includes both bad and good units, and also the units that are on the whole probe. So not just restricted to your region that you were interested in. So what we need to do is basically first limit our clusters or our spikes by those that were labeled as good, so passing our quality control metrics. So in this case, this is indicated in our data by this clusters label. So any cluster that has a label of 1 indicates that it passes our 3 quality control metrics and is good to use. And then we can also restrict by only the clusters that pass in our region of interest, so in this case, GRN, by restricting, by using the acronym keyword here, and basically restricting our clusters to those 2 conditions. In this way, we can then restrict our whole spike sorting data set, which probably contains something like 2 million neurons to much fewer neurons that are just matching our criteria, and then go forward with our analysis. Okay, so in the next section, we've now loaded in our trials data, our spike data, and prepared them in a way that we need for this analysis that we're doing. And what we can then do is now go ahead and go through that kind of workflow that we were showing earlier. So steps A and B for how we can compute the PSTH and then stack the PSTH across the different neurons. So I should emphasize that in this these code snippets, this is just pseudocode. So the functions for compute PSTH, stack PSTH don't actually exist. I'm just showing you how you would then add in your code here to then do the kind of actual crunching of the numbers itself. So for this analysis, we basically want to loop over all of the clusters that we have found on our probe. And we can then get this high dimensional array where we have these PSTHs over all of the units that we have for this single probe insertion. Yeah, and then we can then go ahead and compute the PCA. So we can compute this on the left PSTHs and also the right PSTHs, get our trajectories, and then basically we can pass this into a function called compute distance, where we will then see how the distance varies across the trajectories. So up till now, we've basically just done our analysis on one single probe insertion. So we've set it up, we've loaded in the data, we've prepared our data, and we've also computed the variables that we're interested in. So once we've done that and confirmed the results seem reasonable, what we can then do is basically extend our analysis. So in this case, all we're going to do is just loop over those 17 insertions that we were given in the first instance, and basically just keep extending this analysis such that we can then have multi-session data, which then will make our results more significant and more kind of powerful in this way. Okay, so that's the end of this section where, basically I've shown you how you can use the tools and also refer to many different resources that are available. So hopefully you can have more familiarity with what's available and how to get started using the data. So I just want to walk you through a couple of the resources that we have available, and then I'll take any questions. So first, I just want to emphasize these 3 tutorials that we have here. So these are Colab tutorials that we've written. So we presented these at some other courses. And they're basically work-along booklets that you can use that introduce you to the ONE API and the different dataset types. So when getting started with our data, I really recommend you to particularly work through the IBL ONE tutorial, because this will give you a feel for how to search for data, load different types of data, and we also show how to load video data, DLC data, and other things as well. There's also this IBL brainwide map analysis collab, which I've actually taken inspiration from this trajectory analysis, but it also shows how you can do some simplified encoding and decoding analysis on the brainwide map data. So the rest are kind of other things for your reference. So we have the IBL website, the visualization website, which I think you were exploring yesterday as part of the assignment. And we've also linked to both the brainwide map paper, where we've taken these results from. And alongside that, we also have a technical paper that describes in more detail the processing pipelines that we've applied, for example, the spike sorting algorithm, the DLC pipeline that we've used, and also some of the quality control that we've done on our data to make sure it's fit for public use. So you can explore that to see how we prepared our data. Next slide, please. And yeah, so I've referred to a lot of these documentation as I was going through, but here are the links for example, the description of all of the data sets that we have available, the loading data documentation, and also the O&E documentation that has more information about how to use the API. And yeah, finally, I just want to say if you have any issues, these are kind of the 3 places where we ask you to seek help. So the first is we have a forum, so we subscribe to the Neurostars forum, and if you post a question there with the tag IBL, we can do troubleshooting there. You can alternatively post an issue on IBLM, or if you have any general questions, then you can email info@internationalbrainlab.org. We recommend for kind of API usage and kind of technical problems to use the first 2 things, and the third, the info@International Brain Lab, to be more general questions about the data or other publications. So with that, I'd just like to thank everyone in the International Brain Lab for this amazing effort and all of our sponsors. And again, I think we've emphasized one more slide over, Miles, before, but this work really wouldn't have been possible without like years and years of hard work from these experimenters. So we really want to thank them all for all their hard work of collecting this incredible data set and making it available for everyone to use. So yeah, happy to take any questions.